selection inventors. Like he really was, him and Jerry Joyce and I think Larry Gold, the three of them were sort of the big three in selections, so starting Aptamers back in the day. But the main inventors of the strand displacements and circuitry are Andrew Tuberfield, who was at FDN
Yeah.
when we were there, which was really great to meet him. And then the other two big dogs in that are Eric Winfrey and Georg Selig. And I'm going to touch on that a little bit. And Bernard Yerk, that's another big name in it. Actually, Georg Selig, after I've been trying to pull either Winfrey or Selig to give a talk at our university for 5 years now. And Georg Selig finally got picked this year on the day that my postdoc ends. So I was telling my boss, I was like, I don't care. I want to be at this talk.
When is it gonna be?
I think it's March 30th. It's a
Okay.
Tuesday. So yeah, I mean, if I can, maybe I can share the link with you guys if you want to listen in.
Well, I would definitely love to.
If it's allowed, I'll let you know.
That would be nice. Thank you. Yeah,
Yeah.
that would. Antonio, I guess we can get started.
Yeah, I will give a little introduction to the talk. So yeah, today we have Adam Kabza as an invited speaker giving a talk about chiral DNA nanotechnology. Adam completed his undergraduate studies at Tulane University in New Orleans, Louisiana in 2014. Having a bachelor degree in chemistry and math, he matriculated to Texas A&M University and joined Dr. Shipansky lab, where he has worked on chiral nucleic acid processing, including 2 published works on selecting an L-RNA aptamer and on communicating sequence-specific information between 2 enantiomers of DNA. Despite the significant progress made in the last 2 decades in the field of DNA nanotechnology, the field has remained locked with the homochiral paradigm. Therefore, I found it extremely interesting to listen to a talk by Adam about different enantiomers and how they contribute to this field. So Adam, please share now your work and opinion on the potential of L forms of nucleic acids in the applications such as bioengineering and nanomedicine. The podium, or better to say, the screen is yours now. Thank you.
Awesome. Thanks, Antonio. That was a very nice introduction. All right, let me make sure I share with you guys the right thing here.
I guess we can do it. Yeah, right.
All right, and can everybody see that?
Yes.
All right, and then let me give myself a laser pointer. All right, excellent. So, all right, yeah, thank you, Antonio. Yeah, so I'm Adam, and today I'm going to be telling you guys a little bit about heterochiral DNA nanotechnology. Specifically, there are sort of 2 overlapping categories, the sort of structure-specific interactions and sequence-specific interactions. And I'm going to break up the talk into those 2 segments. And then at the end, I'll talk about a little bit how they overlap and why that's important and some of the work we've done at that interface. Come on, what's going on? No. Sorry. There we go. All right. So yeah, I'll begin by introducing L-nucleic acids and arguing why they're better chemical modifications than sort of the classic modified oligonucleotides. I'm going to talk about aptamers and L-aptamers and precursor microRNAs, which are the things we target with our aptamers in our lab. I'll then talk about the mirror image selection of a chemically modified L-RNA aptamer, which was my first publication in this lab. And then that will sort of constitute the structure-specific half of my talk. And then in the second half, I'll tell you about heterochiral strand displacement reactions or circuits, which I will use circuitry and strand displacement interchangeably in the case of this talk. And then I'll talk about my LDNA signal amplification circuit, which will be based on the catalytic hairpin assembly. And at the end of this, I'll show you guys what myself and other people in my lab are working on in this field and where it's going and where we think it can go. So, all right, why do we modify nucleic acids? Well, they increase nuclease stability when delivering exogenous nucleic acids to living systems. Obviously, DNases and RNases just shred everything because they think it's a virus, probably. So that's one of the sort of pros of having a chemical modification. Some of the cons, and I think the most prominent sort of con, is that the kinetics and thermodynamics of base pairing are pretty severely affected by incorporating chemical modifications. I'm going to argue that LDNA is a superior modification to these classics such as phosphorothioate, 2'X, and all of these other things, with an exception being the peptide nucleic acid, which will come back around in the second half of this talk for the sequence-specific heterochiral interactions, and we'll see why that's important then. So L-oligonucleotides have slightly different chemical properties being enantiomers, but almost identical physical properties. So they have identical solubility as their enantiomer, and their hybridization kinetics and thermodynamics with themselves, so L with L obviously, are the same as the natural D stereoisomer. Given the stereospecific environment of biological systems, L-oligonucleotides are effectively invisible in these contexts. And one really key property here that can be viewed as either a pro or a con, which I think is really an advantage more than a disadvantage, is that L-oligonucleotides do not base pair with D-oligonucleotides. And there are obviously examples where people have forced some sort of quasi-base pairing, but for the sake of argument, it's not standard Watson-Crick. So, all right, so yeah, we'll just dive straight into the first half of this talk about the structure-specific interactions. And here I'm just going to tell you all about how we go about selecting L-aptamers against natural RNA targets. And yeah, so aptamers are single-stranded oligonucleotides which bind a specific ligand. Just as some background, it's derived from the Latin of aptus, or to fit. We generate aptamers by a process known as in vitro selection. And in our case, it's a mirror image in vitro selection. And I'm going to explain a little bit about how that works. So typically you take a library of 10 to the 15 molecules or so, which are dRNA molecules. They need to be in the native stereochemistry to be able to manipulate them with molecular biology methods. We incubate them with a target ligand that's biotinylated. Anything that binds is pulled down with streptavidin-coated magnetic beads. We wash the supernatant of this to get rid of anything that doesn't bind. Then once we have sort of isolated these binding oligonucleotides, we elute them, amplify them through reverse transcription and PCR, and transcribe them into a new library of dRNA molecules, which is hopefully a enriched library at this point with an overall higher binding affinity for the target. So some of the limitations of D-aptamers are, like Antonio mentioned at the beginning, they're poor biostability. They have a lot of off-target effects since they can hybridize with any endogenous nucleic acids, of which there are obviously a lot, especially in the nucleus. If you do modify them post-selection by adding, for example, like a locked nucleic acid or phosphorothioates at the terminal ends, you can affect binding affinity and specificity quite dramatically, and that takes a lot of post-modification optimization to make it work effectively. And selecting aptamers that are D stereochemistry, the native stereochemistry, against D RNA targets, you basically are just selecting for complementary sequences because it's the easiest and most efficient route for two oligonucleotides to bind. So it's very challenging to get a D aptamer that binds a D oligonucleotide target through structural specific interactions. All right, so now how do we do a mirror image selection? Is basically done through a process called excuse me, sorry. Mirror image selection we do by synthesizing the L version of our target and selecting an aptamer, which is a D RNA library like I showed before, against this synthetic L target. And then we switch at the very end. Once we've isolated an aptamer that we know binds, we synthesize it in L, and this sort of property known as reciprocal substrate specificity makes it so that the L aptamer will now bind the D RNA target. So in my case here, I'm selecting an aptamer that also has a chemical modification, this amino allele uridine here. So it has this positive charge. Our motivation for this is obviously to reduce the negative charge repulsion between the library of polyanionic oligonucleotides and the target, which is also a polyanionic polymer. The advantage of this amino allele modification is that the T7 RNA polymerase, which is really standard for doing transcriptions in the lab, accepts it as a nucleotide triphosphate. So it's very simple to incorporate into a library. So why do we like to target precursor microRNAs? And this is just a diagram sort of showing the biogenesis of precursor microRNAs. So usually when genes are transcribed, there's this, the 3' UTR or untranslated region that typically contains a decent amount of these hairpin-like structures. These are called primary microRNAs. They're processed by DROSHA and DGCR8 in the nucleus to become precursor microRNAs. And once these precursors are exported into the nucleus, they're processed further by dicer to form the mature microRNA. And microRNAs are involved in a lot of attenuation of gene expression and modulation of gene expression. And the overexpression of or suppression of certain microRNAs is common in numerous disease states, ranging from cancer to just a lot. And specifically a lot of different cancers, I think, is really sort of the main issue there. So what we try to target are these precursor microRNAs, which are not mature yet, and they don't modulate gene expression. Also, because they're a hairpin still, they sort of have enough structure for an aptamer to sort of bind through these tertiary interactions and kind of have that sort of lock and key mode of binding. Yeah, so just a little bit more detail on this. These precursor microRNAs bind dicer, it chops them near the stem loop region here, and releases the mature microRNA duplex, which is loaded onto the Argonaut complex. The antisense strand is thrown away, and an MI risk complex, RNA-induced silencing complex, is formed, which then targets mRNAs that are complementary or partially complementary with the microRNA sequence. And so this leads to mRNA silencing. All right, so I just wanted to show again sort of our selection scheme, except now I'm just showing you that this target here that I was talking about, we made this an L precursor microRNA. And so again, we do this whole series of steps and repeat it numerous times and get an aptamer. So this is the sequence of our aptamer. And also, I guess, before I continue, any sequences drawn in black are D oligonucleotides by convention for us. L is blue. So that's just so that you guys can keep track a little bit better. So yeah, we found this aptamer. I believe it had either 12 or 13 amino allele uridines, which sort of showed us that in fact the amino allele was playing a big role in binding the target. We did a KD analysis, obviously. So I think it ended up being about 100 nanomolar, or excuse me, it was about 1 picomolar binding affinity for the D aptamer, whereas the L aptamer was about 100 or maybe 50 nanomolar, 100 nanomolar. I'm so sorry. I can't see on the screen. I think it was 5 nanomolar, yeah, 5 nanomolar KD for the L aptamer and about 900 picomolar or 0.9 nanomolar for the D aptamer KD. Now, theoretically, the binding affinity should be exactly identical. However, it's really difficult to produce as good of quality RNA or DNA synthetically on an instrument as it is for enzymatic production of oligonucleotides. So obviously this dRNA aptamer has been transcribed in this case by enzymes. And so we think that that probably is what contributes to this discrepancy in binding affinity. Not that it's very high, in my opinion. I don't think it really has that big of an effect. Down here, I just show the specificity of our aptamer. So these are other precursor microRNAs, 17, 28, 21, and 155. I believe, if I recall correctly, 17 and 20A are actually from the same mRNA 3' untranslated region as 19A. So even within the same family or cluster of microRNAs, this aptamer is specific. And then this 21 and 155 are from different mRNA transcripts. Again, just showing that we are hyperspecific for 19A, excuse me. All right, so then to prove or attempt to prove that these aminoallele uridines are really necessary for binding, we took the D form of this aptamer and slowly started replacing the aminoallele uridines with C or cytosine nucleobases. And so basically any UA base pair was replaced with a CG base pair systematically. And again, showed that basically just deduced which aminoallele uridines are required for binding. So the ones in this M1 mutant or the P1 stem, we are not necessary for binding. As you can see, this mutant bound the target. We expect that this P1 stem is strictly there for structural reasons, not for binding purposes. So basically just the fact that the stem exists allows the rest of the aptamer to fold into whatever configuration it needs to bind. And then I can't see behind the camera. Let me see, can I, yeah, there we go. Okay, yeah, and now I can see the end of my screen. And then yes, again, these M8 and M9 mutants here in this P3 stem also were not required for binding. Again, just to us, that says that this stem is basically there just for structural reasons. All right, so once we did this, we needed to check, do we actually inhibit dicer? Because that was sort of the whole point of this system. So we started by doing what's called a footprinting analysis. So as I showed you earlier, dicer cleaves sort of at these 2 points just adjacent to the loop region of the precursor microRNA. And a footprinting experiment is basically where you have the target bound to the aptamer and the target is radio labeled, and we just incubate it at high magnesium concentrations and slowly allow it to digest itself through 2', 3' hydrolysis, backbone hydrolysis. And then we run it down a gel and we check for differences between this footprinting gel when there's an absence or presence of aptamer. So in this lane, there's no aptamer. In this lane, we have aptamer, and you can see that sort of these bands change intensity quite drastically between the absence and presence of aptamer. And we can map this by doing a RNase T1 digestion, which cleaves only after G residues. And so this allows us to sort of map where within the molecule here our aptamer is binding. And so when we mapped this, it shows, like I've highlighted in red here, the bases whose intensity changed the most. And we can clearly see that it's all around the loop region of this precursor microRNA. So that was a really good thing to see. There we go. Can you hear me? Sorry.
Yes, yes.
Okay, awesome. Yeah, no, it's okay. So yeah, once we sort of determined that it binds the loop region here, we were pretty confident that dicer will basically just bounce off from binding. And we did a dicer inhibition analysis. So here I have precursor microRNA 19A without dicer, with dicer and no aptamer. So this is basically how much it gets cleaved in the absence of any inhibitors. And then as I increase the concentration of the aptamer, we get essentially full inhibition of dicer at around 50 nanomolar of aptamer concentration. And I repeated the same experiment against precursor microRNA 21, which is another precursor microRNA processed by dicer, and at no concentration of aptamer did any of the inhibitions change. So we calculated an IC50 of 4 nanomolar, which is obviously quite good. And I believe, yeah, that was pretty much the end of the whole paper. That's what we published on. So yeah, so we developed an LRNA aptamer, specifically a chemically modified one, which this was the first chemically modified LRNA aptamer. My boss, Dr. Strapansky, has a couple of aptamers from his postdoc at Scripps. We demonstrated that the modifications necessary for type binding. We characterized the binding modality with the footprinting analysis. This isn't very strict characterization, but it's the best we can do without crystallography. And we demonstrated the ability of this aptamer to inhibit dicer processing of the precursor microRNA target. So that will conclude sort of the first half of my talk, where we talked about these structure-based interactions. And I guess, yeah, I should mention that, because our L-aptamer can't hybridize to the D target, it is all a tertiary structure-based interactions. At most, there's probably some quasi hydrogen base pairing like bonding between the two structures, but nothing that we can say definitively. So for the second half of this talk, I'm going to talk about how we have made it so that despite the fact that L-DNA cannot hybridize to ddNA or RNA, how did we achieve sequence-based communication between the enantiomers? I'm going to follow it up with a signal amplification circuit based on this. And then at the end, I'll tell you about what everyone else in my lab's been doing. So yeah, so now we get on to the heterochiral circuitry. And this is where PNA makes an appearance again. So yeah, and just to reiterate, D oligonucleotides are going to be black, RNA is going to be red now, LDNA is blue, and achiral PNA will be green. And this will be consistent throughout the rest of the presentation, except here, because I stole this figure. So yeah, basically, this is just the original figure from Andrew Tuberfield and Bernard Yerkes collaboration. And this was published in 2000 in Nature. And this was the first example of strand displacement reactions being used to make sort of dynamic tools with the oligonucleotides. In this case, they've made a tweezer, a molecular tweezer. And the readout of this was based on FRET between TET and TAMRA. So in the first step, a fuel strand here where the green here is complementary to the green in the tweezer, hybridizes and begins to, and the blue on the fuel strand is complementary to the blue on the other end of the tweezer. And so this locks the tweezer, brings the TET and TAMRA dyes into proximity, and allows you to generate a FRET signal. And then the fuel strand has this sort of orange domain left dangling as a single-stranded region. This is called a toehold domain. And that allows us to input the full complement of F or F prime and peel away the fuel strand, generating a waste duplex and reopening the molecular tweezer. So this was just sort of the first example of anyone being able to achieve dynamic strand displacement cascades with oligonucleotides. Georg Selig and Eric Winfrey formalized this 6 years later into true sort of nucleic acid circuitry, where they abstracted these sort of strand displacement events to become to follow Boolean logic and Boolean circuit logic. So essentially 0-1 logic of inputs, outputs. And in this case, they have a 2-input gate, and the first step is G in, binds to this toehold here and removes the G strand, opening up this purple toehold domain, the second toehold domain that was initially blocked in the first duplex. The next input, F in, comes in and will bind to this, it's called the substrate strand is the one that the input binds to, and the incumbent strand, or this E out strand, now falls away, and it again has yet another toehold here that was originally sequestered through duplex formation. And in this case, they even have this sort of this loop protruding from the duplex, allowing your downstream oligonucleotides to have even more sequence space available to them for downstream inputs. And they just sort of show this fluorescently with a fluorophore quencher module. When they input both inputs, quite quickly, you generate a fluorescent signal as the fluorophore is released from the quencher. And so this is sort of just the general outline of how these strand displacement circuits work. I'm going to be following very similar notation to everything. And the reason that this is so important is that we can imagine each strand displacement duplex to be a logic gate analogous to digital circuitry. And instead of having a high or low current be your 0-1 input or output, it's the high or low concentration of an oligonucleotide species that act as your 0-1 binary bits. And obviously there are vast implications for this, given the fact that your genetic profile and genetic expression is all based on transient oligonucleotide species, mRNAs, microRNAs, any of these types of things, and even just the euchromatin, heterochromatin, like how densely compacted your chromosomal DNA is. And because these strand displacements work based on sequence complementarity, it allows you to directly interface engineered logic circuits with living organisms or living cells. And so basically we now have sort of our own digital tool for plugging into a cell and either getting readouts or modulating certain things. Now there are obviously a lot of implications that make doing something like this difficult, but the whole I guess dream of this field is to have oligonucleotide inputs. You get a biocomputation and you output a fluorescent readout, like a diagnostic readout or a therapeutic release, or both. You can all of this is modular since it's Boolean logic, so it's very easy to sort of nest egg all of your different logic needs, I suppose. All right, so what did we do? we basically figured out a way to make L oligonucleotides sequence specifically interact through peptide nucleic acid with D oligonucleotides. And the way that this works is that PNA has an achiral backbone. It has this N2 aminoethyl glycine backbone with no chiral carbons in it. The bases protrude from the amine group here, and they will indiscriminately hybridize with D or L oligonucleotides, and specifically they will take on the helical shape that is preferred by the oligonucleotides. So when and this will be important, and I'll show why a little bit later on. So if PNA binds to an LDNA, it will make a left-handed helix. If it binds ddNA, it makes a right-handed helix. And obviously, this is really, we think this is important because now you can set up your entire logic circuit in L chirality and just have very specific points of interaction through this PNA module to the native stereoisomer. And this way you basically minimize any off-target hybridization effects to endogenous nucleic acids that you don't want it to hybridize to, since it's in the L stereochemistry, as well as obviously the basically complete nuclease stability of L nucleic acids. And in addition to that, I don't think I mentioned before, but L oligonucleotides also have almost no, elicit very little immunogenic effect in living systems. So there's a whole host of sort of advantages to using LDNA. So yeah, so we basically, the same principles that I just discussed can be applied to PNA-DNA duplexes. So the same as a DNA-DNA duplex. There are obviously kinetic and thermodynamic parameters that need to be assessed. PNA hybridizes more strongly with its complementary oligonucleotide strand, since it has a it's a neutral backbone. It's not ionic, so it does bind a little more tightly than the analogous oligonucleotide would. And so there are sort of two scenarios that can be had from, there are two general strand displacements. I'm going to talk about one here and another one a little bit later. The first is you have the PNA is the incumbent strand. And so either a ddNA can hybridize to the toe-hole domain. So any starred domain is complementary to the unstarred domain. So here T will transiently nucleate and hybridize to T star and begin strand displacing the PNA. And below it, between the mirror plane, we have the same thing happening in the L stereochemistry. And in both cases, you generate an identical but stereochemically inverted waist duplex and output an achiral PNA, which downstream can go on to interact with oligonucleotides of either stereochemistry. And so we call this a racemization gate. And the first circuit we designed based off of this was our racemization circuit. And so this is going to be a lot of duplexes. I apologize, but I'm going to try and make it as clear as possible. So basically, the way you can look at this is that everything happening above and below the dotted line is identical. It's just D stereochemistry up above, L stereochemistry down below. And I'm just inserting this PNA along the mirror plane since it's achiral in the steps where it has an option to go down the L path or the D path. And so just like I showed in the previous slide, we first have this displacement of the incumbent PNA to become freed. Now, this PNA can go on to interact with duplex B1, so either the DB1 or LB1 down here. And this generates the output strand 4', 3' in either case. And this 4', 3' is designed to go on to open a reporter duplex, which in the case of the L stereochemistry has a Psi5 fluorophore with a quencher. And in the D stereochemistry, we have a Psi3 fluorophore with its quencher. And when this output strand interacts with the reporter duplex, it releases the fluorophore and generates either a Psi3 or a Psi5 signal. And we specifically chose Psi3 and Psi5 in this case to have orthogonal fluorophores so that in real time we could track exactly which half of our circuit is being activated. And the reason that this was important to us is because you could basically mix and match all different DNL components and generate lots of different pathways. So for example, we can have these duplexes, this DA and DN duplex, LB duplex, and L reporter present and D reporter present. But if we did not include this DB1, and we put an input in, we would track only fluorescence of the L reporter, despite the D reporter being present. And you can do the opposite, going from L into the D stereochemistry. And also, what I thought was probably the coolest experiment we did, if you have all components present, except for, say, the L input, if you put 2 equivalents of D input, and 2 equivalents of this DA1, which would then release 2 equivalents of the PNA, you will get full activation of both the D and the L reporters. And I'll show you what that looks like in just a moment. So in the first case, sort of like I mentioned, we have pathway 2, and there were many pathways in our actual paper, but I'm just going to focus on the ones that I want to talk about. So pathway 2 is where we have a D input, DA1, LB1, and L reporter, and D reporter present. In all of these cases, both reporters are present just to really show the orthogonality. And so you can see here the Psi3 or the D reporter is never activated at all. And in fact, there's even very little leak in this circuit. Whereas the L reporter, because the LB1 duplex is present, obtains full signal over the course of about 2 hours. And we show the same in the inverted stereochemistry. And moving on to the experiment I described just a moment ago, if we input 1 equivalent of D input, but have all the downstream components present, both circuits will activate to exactly 50%, which is shown here in the blue. And when I put 2 equivalents of this D input and DA1 for 2 equivalents of PNA, both circuits halves will activate to full activation. And so we thought this was really cool. And obviously it was published in JACS, which we were really excited about. And this was all, I should mention, this is all done with my colleague, Brian Young, who's now a postdoc at Stanford. So, one of the things I mentioned previously was how there's a second sort of general reaction of these heterochiral strand displacements that's not a racemization, and it's the chiral inversion. And this is where the PNA is the substrate strand, and the incumbent strand can be an oligonucleotide of either stereochemistry. In this case, we want L. And here, when a D or an L input are included, which are complementary to the PNA, basically a D can displace an L strand. And so now sort of the reason that this is sequence-specific communication between the oligonucleotides of opposite stereochemistry is the sequence domains 2, 3 on this D oligonucleotide are identical to this L. And so, you have this direct transfer of sequence-based information between stereochemistries. And what's really cool about this reaction, this was sort of the reaction that Brian worked on in this paper. I worked on the racemization gate, and he worked on this chiral inversion gate. What's really cool about this, and a really interesting caveat that we probably should have expected, but we didn't, is that if you look here, so with no inputs in this red, we have no activation. When an L input is included to displace an L oligonucleotide, we get really fast activation here. When the D input is placed to displace this L, we get a really slow, relatively much slower activation than the L. And so there's basically, we realized that because this duplex is going to be a left-handed helix, there's a basically kinetic and thermodynamic penalty to doing the branch migration. So basically for this D to start displacing the L, it has to rearrange the entire the entire structure of the helix from L to D. And so this causes this kinetic penalty. And I'll talk a little bit more later about what else we've been doing with that. But that was just a really cool sort of unexpected or unforeseen rather result in this circuit. So I just thought I'd mention that because I thought it was really interesting. So kind of getting on to the final portion of my talk, we want to be able to translate information from D to L do this information processing in L, and then switch back either with an L diagnostic, like an L reporter, or switch back into the D stereochemistry and release a D therapeutic, such as an antisense oligonucleotide, Aptamer, et cetera, that can go on to interact with things inside of a cell. We decided to start with generating a signal amplification circuit for the detection of microRNAs and by extension other single-stranded RNAs. To do this, we pursued making an L-catalytic hairpin assembly, or CHA as I'll refer to it. So in this case, again, we move to L, catalytically amplify a signal, and generate a fluorescent output. So this will, again, be based on this inversion gate, not the racemization gate, because we need to go directly from a D microRNA input to an L output, which will then activate our catalytic hairpin assembly. All right, so this is probably the most brutal circuit I have to show you guys, so bear with me. We have D microRNA comes in and displaces this L155 strand. This strand now goes on to activate the L hairpin 1. L hairpin 1, once opened, interacts with L hairpin 2. When L hairpin 2 hybridizes to L hairpin 1, it displaces again this L out 155 strand. So for one singular displacement of the L strand here, you get multiple turnovers of this catalytic hairpin assembly, which generates this duplex of the two hairpins. And that duplex now has this 4-7 domain, which is complementary, again, to a fluorescently labeled reporter duplex. And so when these hairpin duplexes are formed, they can start to activate the reporter and generate a signal. And so we did this reaction in a lot of different contexts. So first, we just basically showed that the full LCHA works with a D input. So in this case, I have incubated the circuit for 2 hours with no input, just to basically show that there is not a tremendous amount of leak. And then at the 2-hour mark, I initiated the CHA by spiking it with this D microRNA 155. And so basically the different line colors just correspond to different concentrations of input of D mirror 155. Obviously, like I said before, the chiral inversion gate has impeded kinetics to displacement. So when you compare this reaction to the version where I have an, excuse me, let me just make sure. Yeah, to the version where I have an L155, the kinetics are a little bit faster with the L155, and this is more noticeable in other experiments that don't include FBS, which I don't show here, but you can take my word for it if you trust me. One of the things we wanted to show was that our circuit will work really robustly in the presence of 10% fetal bovine serum, which mimics sort of harsh biological environments. It's got lots of nucleases and stuff. The problem with this, and what made it extremely difficult to sort of show that this works in the case with a D microRNA input, is when I would add D microRNA to a reaction containing FBS, it would just annihilate the input before it could even activate the circuit. So for all of the reactions in FBS, I had to use just the L version of the 155 as a stable constant concentration input. Oh yeah, so here you see the full DCHA with the D155. You can certainly see, I think, the kinetics of those reactions are much faster and it behaves much better when a D input is used on a D inversion gate, since it's not actually inverting stereochemistry. But then when we do the DCHA in the presence of FBS, we see that it basically just degrades, and there's no change when I add initiator. There's a small bump here. It's from a concentration change as I increase the volume of the reaction. And so, we were really happy with these results. Obviously, they worked in FBS, whereas the D didn't. We also did an experiment where we attempted mismatch discrimination. So here I've basically taken the one of the 4 nanomolar input concentrations and shown that at one of the lowest concentrations where we could still detect a significant signal change, if we have mutant 1 or mutant 2, which basically just in the toehold binding domain here, where this 1 binds to 1 star, I've just made either 1 single point mutation or 2. We see no activation of the circuit, or basically none, relative to just the background leak when no input is included. And this was also done in FBS with an L version of all of these things. So yeah, so basically overall, we developed a novel DNA strand displacement reaction capable of communicating sequence-specific information between enantiomers of nucleic acids. We can discriminate against incorrect input sequences, which is also something that has been shown, obviously, in all of the homochiral strand displacement literature that's out there. And we demonstrate that our reactions are superior for use in harsh biological conditions. And yeah, we can detect trace oligonucleotide species in these harsh media. So where do we go from here? So these are just my colleagues. This is my colleague Brian Young and Nandini Kundu, who's also a senior graduate student in our lab. And they basically, as I mentioned before, he worked on the chiral inversion gate of our original manuscript. And so the two of them together have been working on and finally recently submitted the characterization of reaction kinetics of heterochiral strand displacements, specifically for the chiral inversion gates. And they basically just follow, Eric Winfrey has a paper in nucleic acids research that's like the Bible of strand displacement kinetics, and they basically just tried to replicate a lot of the experiments in that work among the new experiments that are able to be done in this heterochiral context. And you can see here pretty clearly that in the homochiral, you kind of get this nice spread of kinetic activity with as you change the toehold domain from 2 bases to 10 bases. Obviously 10 bases being in green here, it's very fast because you can nucleate very easily without falling off. And in the D case, you have activation of the circuit, at even 2 and 4 nucleotide toeholds, and then at 6 you get this big jump, and then it keeps going. Whereas for the inversion, until you get to about 8 bases, you have really, really slow kinetics and basically just no reaction taking place. This can all be quantified, toehold length versus log K. And the homochiral is faster, obviously, overall in all contexts. So another publication that came from this was some work that Brian did, and this was to use chimeric oligonucleotides. So basically oligonucleotides that we synthesize in-house, where a portion of them is in the D stereochemistry and a portion is in the L. And we can again sort of get these chiral inversions. This ddNA input will displace this chimeric LD-DNA output, but this L portion is now made to bind the reporter domain. And this is just basically another construct where the displacement of this strand here will allow this all L oligonucleotide to go on to interact with the reporter. And so you could argue that this is even more beneficial than a chimeric displacement because it's still subject to off-target nucleation or hybridization and nuclease degradation. Some of the work I've been working on, and this is unpublished results, so don't tell anybody, but I just think it's really cool, so I wanted to tell you guys. These are these quasi-heterochiral ligations, and this is towards constructing large L oligonucleotides. And so basically what I've done is I've found sort of minimal sequence requirements in terms of putting D oligonucleotides at the terminal ends of long L oligonucleotides. And I found that different ligase enzymes that are commercially available accept this D domain and will ligate your two long L strands together. This is pretty nice, and it works honestly really well depending on how many D oligos you're willing to incorporate into your structure. But we can get down to, I think the best we've done so far is 4. So you need 2 on this end and 2 on this end for the ligases to ligate with any semblance of efficiency. And you know, we've constructed a couple of L oligos that are functional, and we can ligate them and they're still functional. And so I think this is pretty cool. We're probably going to publish on it pretty soon. Another one of the students in our lab, Wenrei, he has been working on the cellular work with all of this stuff. So he designed a PNA LDNA duplex that detects microRNA input and releases this L-mango aptamer, which can't fold while this duplex is stabilized. So once the L portion of this duplex is released, the thiazole orange that the cells are transfected with can bind the L-mango aptamer and generate a fluorescent signal for live cell imaging. And finally, sort of more in the structural domain of all of this stuff, Charles Deckard is another, well, he's a postdoc also in our lab still. He works more in the chromatin side of our lab, but he and John found that this PRC2 protein, I don't remember what it stands for right now. I'm not a protein chemist, but basically PRC2 is polycomb repressive complex. That's what it is. It's known to be recruited for basically condensing and decondensing chromatin to modulate genetic expression. And it had been reported previously that a DG quadruplex, which binds PRC2, can inhibit its activity. And so our argument was that, excuse me, the argument was that even though D and L oligonucleotides have opposing stereochemistry and are going to have, you know, different overarching stereochemistry even within the context of a G quadruplex, because a G quadruplex could be viewed almost as a cube. You know, if you draw a diagonal through a cube, it's the same diagonal no matter which direction you come from. And so basically we thought that the G quadruplex will still bind just because it basically is binding PRC2 like a cylinder is fitting into a keyhole or whatever. And so he basically showed that this does in fact happen when you have PRC2 that's binding this RNA complex, which is a giant smear up here, versus free chromatin, which is shown down here in a control. As you increase the concentration of this LG quadruplex, you move back from this smear into free chromatin, basically implying that we are inhibiting PRC2 with this LG quadruplex. And then he did another set of experiments that I thought were really cool that I'll show here. So PRC2 is recruited by long non-coding RNAs, in this case hot air, which also bind chromatin and recruit PRC2 among other proteins to do different things to the chromosomal DNA. And whenever Charles exposed PRC2 to this LG quadruplex, he basically could knock down the PRC2 hot air complex as the concentration increased. So this just sort of shows that even though a lot of these interactions are very stereospecific, you know, we can find sort of examples and exceptions that make the rule almost, if you will, through this sort of G, in this case, it's a G quadruplex binding. And so I think that's pretty much the most of my talk. Yeah, so we expanded the chemical toolbox of lRNA aptamers by making a chemically modified one, made a new DNA strand displacement reaction, detected microRNAs with that. I guess you guys can ask me more questions about this, but I didn't really touch on it too much, but I think and we think that these types of tools can be utilized ubiquitously in nucleic acid nanotechnology. You basically just have to switch chiralities and buy some PNAs. And we've progressed to in vivo applications of these technologies, as well as the kinetic characterization, and also applied them to some of these chromatin projects in our lab. So with that, this is our group, everybody, and our funding sources: NIH, Cancer Prevention and Research Institute of Texas, and the Welch Foundation. And that's it. That's my talk, guys. If you have any questions, I would be happy to talk about some LDNA stuff. So,
Adam, thank you so much for your talk. Despite the complexity and amount of the information